what if the solutions to like some of the biggest challenges we're facing right now,
Yeah.
like climate change, all the diseases we hear about, the massive amount of plastic everywhere,
Hmm.
what if those weren't separate problems?
Right.
Like huge mountains to climb, one by one.
Right. Yeah.
What if they were all connected?
Uh-huh.
Pieces of a puzzle, but the key to solving it was like almost too small to see.
That's a really powerful way to think about it,
Ah.
and honestly, it's closer to reality than you might think.
Okay, so how are all these global problems connected at a tiny scale?
Recent breakthroughs in science are suggesting that this interconnectedness is more real than we ever thought.
Hmm.
It all started with solving a fundamental problem in biology.
Really?
But the ripple effects are spreading out into areas that, might seem completely unrelated at first glance.
So what was this central puzzle that was holding back biology for so long?
Well, it was this incredibly tough problem of figuring out the 3D structure of proteins. It sounds simple, but it's been called the Fermat's last theorem of biology because it was so hard to crack.
Fermat's last theorem of biology. Wow, that sounds pretty intense. How long were scientists stuck on this?
For over 6 decades, thousands of biologists were working really hard, painstakingly trying to map out the structure of proteins. They only managed to figure out about 150,000. It was a massive effort, but honestly, it was just scratching the surface when you consider how many proteins there actually are.
6 decades, just for 150,000. That really shows the scale of the problem, right? And then something changed.
Yeah, dramatically. In just a few years, a small team, like just 15 people or so, they used artificial intelligence, AI, to determine the structure of a mind-blowing 200 million proteins.
200 million. That's not just progress, that's like a quantum leap forward.
Right, you said it. So basically, they now have a way to understand the very building blocks of life on this massive scale, all thanks to a breakthrough in AI.
So the big questions for me and probably for our listeners are, first, how on earth did they do that? It seems impossible. And second, why should the average person care about this very specific discovery in biology?
Those are great questions, and that's exactly what we're going to dig into today.
Awesome.
But let's start with the basics. What are proteins and why is their shape so incredibly important?
Okay, let's get the foundation laid. What are these proteins we're talking about?
All right, so think of proteins like long, complicated strings. And the beads on this string are called amino acids. Now, each amino acid has a central carbon atom, and attached to it are an amine group on one side and a carboxyl group on the other.
Okay, so it's like this carbon atom is the central piece, and it's got these two groups attached.
Yeah, and what makes each of the 20 different amino acids unique is this thing called a side chain. Think of it like a little attachment that gives each amino acid its own special properties.
So we've got these 20 different amino acids, each with its own side chain, kind of like Lego bricks with different attachments. How do they then become proteins?
Right, so these amino acids connect together to form the protein string.
Uh-huh.
The amine group of one amino acid can react with the carboxyl group of another one.
Okay.
And this forms a chemical bond called a peptide bond. So basically, it's like linking those beads on a necklace, but this necklace is super sophisticated.
Got it. We've got our protein chain forming, but we keep hearing about these intricate 3D shapes. These chains don't just stay straight, right?
No, definitely not. They coil and fold into all these amazing and complex ways. This folding is guided by a bunch of different forces at the molecular level. Electrical attractions, hydrogen bonds, interactions with the surrounding water.
Okay, so all these tiny forces are pulling and pushing.
Yeah, it's kind of like like a long piece of string
Okay.
with some sticky parts that want to cling to each other and some parts that repel each other.
I can visualize that.
It's like microscopic origami, almost. And the crucial thing is that the final 3D shape is what determines what the protein actually does, its function. It's like a key fitting into a lock. The shape has to be just right.
Okay, so if the shape isn't right, it doesn't work.
Precisely. Proteins are the workhorses of our cells. They're like tiny machines.
Yeah.
And if the parts aren't in the right place, the machine can't function. For example, take hemoglobin. It carries oxygen in our blood,
Right.
and it has this specific pocket that's perfectly shaped to grab onto oxygen molecules.
So if hemoglobin wasn't folded just right, it couldn't carry oxygen.
That's right, and we'd be in big trouble.
So understanding the 3D structure is really the key to understanding what a protein does. But as we heard, figuring out that shape used to be a huge challenge. What did scientists do before AI came along?
Well, the main technique they used was called X-ray crystallography.
Yeah, X-ray crystallography, that sounds very technical.
It is. So first, they have to get the protein to form a crystal.
What does that mean?
It's like a very ordered, repeating arrangement of protein molecules. It's kind of like how salt forms crystals.
Okay, so the protein has to be organized in a specific way.
Yes. And then they blast this crystal with X-rays.
Uh-huh.
And the X-rays scatter in different directions as they pass through the crystal. Scientists collect this diffraction pattern, basically a pattern of spots.
Okay.
And they use really complex math to figure out what the 3D structure of the protein must be to create that pattern. It's like shining a light through a complex object and figuring out its shape from the shadow.
So they have to make the protein crystal and then they interpret its shadow using X-rays. That sounds incredibly difficult and time-consuming.
It definitely was, and still can be for certain proteins. A good example is the work of John Kendrew. He's a British biochemist who spent 12 years figuring out the structure of myoglobin.
12 years for one protein. That's incredible. What made it so difficult?
Well, myoglobin stores oxygen in our muscles. Kendrew first tried to crystallize it from horse heart,
Oh.
but the crystals were too small. So he realized that diving mammals have way more myoglobin in their muscles because they need to store a lot of oxygen. So he got a huge piece of whale meat from Peru.
What? A piece of whale meat?
That's right. And that finally gave him crystals big enough to work with. Talk about dedication.
Going halfway around the world for a piece of whale meat just to get enough crystals. That's a great story. What did this first protein structure look like?
Well, people thought it would be something elegant, maybe predictable with math. But it was actually incredibly complex, almost messy looking. It was even nicknamed the turd of the century.
The turd of the century. That's memorable.
Right. So even after this huge effort, they found that proteins are a lot more complicated than they thought.
And the progress after that, was it faster?
Sadly, no. In the next 20 years, they only figured out about 100 more protein structures. It was a real bottleneck for research.
So years and years for just a handful of new structures, I can see why they needed a better solution.
Yeah, and it wasn't just the time, it was also expensive. X-ray crystallography can cost tens of thousands of dollars per protein.
Wow, so scientists really needed a faster and cheaper way to do this.
Exactly. That's where the idea of predicting protein folding using computers came in. They thought, if we know the sequence of amino acids, can we just simulate how it folds?
It seems like that should be possible. We know how atoms interact, right?
Yeah, that was the hope. Some early successes included things like predicting secondary structures, like alpha helices and beta sheets.
Okay, so they could predict some of the smaller folding patterns.
But the whole 3D structure, that was still a mystery.
Why was that so hard?
Well, it turns out evolution doesn't design proteins from scratch. It kind of tinkers with them over millions of years. It's like a programmer who just keeps adding to a piece of code that works okay.
So it's not planned out from the beginning.
Nope. And that makes it really hard to reverse engineer.
And to show how hard it is, there's this thing called Levinthal's paradox. Have you heard of that?
Oh, yeah. Leventhal figured out that even a short protein with just 35 amino acids could fold in so many ways, more ways than there are atoms in the universe. Even with a super fast computer checking trillions of configurations per second, it would still take longer than the age of the universe to find the right one just by brute force.
Wow. Longer than the age of the universe. So trying every possibility was just not an option. So what did they do?
Well, in 1994, a scientist named John Molt decided to kickstart progress with a competition called CASP, the Critical Assessment of Protein Structure Prediction.
CASP, a competition. So it was like the Olympics for protein folding algorithms.
Exactly. Teams would create computer models that could predict the 3D structure from just the amino acid sequence. Then they would compare their predictions to the real structure. A perfect score was 100. Anything over 90 was considered solved. That was the goal.
So that 90-plus score, that was the holy grail of protein structure prediction. How did the first competitions go?
Well, the scores were pretty low at first, like in the 40s, but one algorithm called Rosetta started to stand out.
Rosetta, I think I've heard of that. What was so special about it?
One of the big things was that Rosetta used distributed computing. They had this program called Rosetta at Home that people could install on their computers, and it would use their spare processing power to help with the calculations. It was like creating a giant supercomputer out of everyone's home computers.
That's pretty cool, harnessing the power of like a global network of computers.
Yeah. And this even led to some funny interactions. People were watching the screensaver of the program, which showed the protein folding, and they thought they could do it better than the computer. They were sending messages to the scientists saying, I think I can fold it better.
Wow, gamers trying to beat a sophisticated algorithm.
Right, and the scientists took this feedback and created a video game called Fold It.
A video game.
Yeah, where players could actually manipulate a protein chain with their mouse, trying to find the best shape.
That's a brilliant way to use human intuition. Did it actually work?
It did. Over 50,000 gamers worked together and actually solved the structure of an HIV enzyme. And their solution was later confirmed by X-ray crystallography. The gamers even got to be co-authors on the scientific paper.
That's amazing. Real scientific progress made by playing a video game. And this brings us to another big name, Demis Hassabis, the founder of DeepMind. He was one of those fold-it gamers, right?
Exactly. He was super interested in protein folding, and he had this AI company called DeepMind. They had already made a big splash with AlphaGo, an AI that beat the world champion in the game of Go.
Oh, yeah, I remember hearing about that. So he was already thinking about using AI for big problems.
Absolutely. After AlphaGo, they had a hackathon where they tried to train an AI to play Fold It.
So it's his experience with that led to AlphaFold.
Yeah, it really sparked his interest in using AI for scientific discovery.
Meanwhile, back in the CASP competitions, the traditional methods were kind of stuck, right?
Yeah, even Rosetta wasn't getting much better. They had more data and more computing power, but the predictions weren't improving.
So that's where DeepMind came in with their AI expertise.
Yep, they created AlphaFold 1. It used a type of artificial intelligence called a deep neural network.
Okay, so they're feeding a bunch of data into this neural network. What kind of data?
Well, they used the amino acid sequence of the protein,
Makes sense.
but they also added something really important: evolutionary information.
Evolutionary information. What do you mean?
So over millions of years, proteins change a bit.
They mutate, right?
Exactly. But for a protein to work properly, it needs to keep its basic shape. So you can compare the same protein across different species and see which parts of the sequence stay the same.
Okay, the parts that are essential for its function, they stay similar.
Right. And you can also look for mutations that happen together.
Like if one amino acid changes, another one nearby also changes to compensate.
Exactly. That tells you those two amino acids are probably close to each other in the 3D structure. So AlphaFold used all this information.
And what did it actually produce as an output?
Well, it didn't directly predict the 3D structure.
Okay.
Instead, it predicted something called a pair representation. It's like a 2D map showing which amino acids are near each other.
So it's a map of the distances and angles between the amino acids.
Exactly. And then a separate algorithm used this map to try and build the 3D structure.
Okay, so it's a two-step process. How did AlphaFold 1 do in the competition?
Well, it definitely made waves.
Did it win?
It was the clear winner, but it didn't get that 90-plus score. It got an average score of 70.
So it was a big step forward, but not quite there yet.
Yeah, it showed the potential of AI, but it needed more work. So they went back and created AlphaFold 2.
And that's the one that made all the headlines. What did they change to make it so much better?
Well, AlphaFold 2, led by John Jumper, was a much bigger change. They didn't just add more data or computing power. They actually redesigned the AI itself to better understand how proteins work.
So they made the AI itself smarter.
Exactly. There were three key improvements. First, they used massive computing power from Google. Second, they improved the algorithms themselves.
Clever algorithms. Okay.
And this was actually more important than just having more data.
How so?
Well, AlphaFold 2 got much better results using the exact same data as AlphaFold 1, just because the algorithms were better.
Wow.
And the third key thing was a new type of AI called a transformer.
A transformer, isn't that what powers things like those large language models, like the one we're talking to right now?
Exactly.
So how does a transformer work? I don't really understand how they can be applied to both language and proteins.
Well, a key part of a transformer is something called attention. It allows the AI to figure out which parts of a sequence are most important. Think of the sentence, the animal didn't cross the street because it was too tired.
Okay.
We know it refers to animal and not street because of the word tired. The attention mechanism lets the AI make those connections.
I see. So it's figuring out the relationships between words, or in this case, amino acids.
Precisely. And they created a special transformer just for protein folding called the EVO former.
The EVO former.
And this EVO former had two parts, two towers working in parallel, a biology tower and a geometry tower.
So one tower for the evolutionary information and one for the distances and angles.
Exactly. And they constantly exchanged information through a bridge. The biology tower looked for conserved sequences and co-occurring mutations, anything that hinted at which amino acids are close to each other.
In the geometry tower.
It calculated the distances between all the amino acid pairs. It even used a clever trick called triangular attention.
Triangular attention. What's that?
It used the fact that in any triangle, the sum of two sides is always greater than the third side.
Okay, the triangle inequality. I remembered that from geometry class.
So if the predicted distances between amino acids didn't make sense geometrically, the AI knew something was wrong.
So they're using basic geometry to help constrain the possibilities. That's really clever.
It is. And then all this information went into a structure module, which actually built the 3D model.
So the structure module, that was the final step.
Yeah. And it had a unique approach. It didn't force the amino acids to be connected in a chain.
Really?
It just placed each amino acid in 3D space based on the predicted distances and angles. The chain structure just emerged naturally.
Wow, that's pretty amazing. So the AI was learning how proteins actually fold in nature.
Yeah, and this flexibility sometimes led to some weird movements in the visualizations. It's like the protein was figuring itself out.
And then they had this recycling process, right?
Yep. Once they had a 3D model, they'd run it back through the EVO former a few times to refine it.
Make it even better.
And the results, as you know, were incredible.
Yeah, they went back to CASP with AlphaFold 2 and blew everyone away.
Yeah, they finally broke that 90-plus barrier. John Molt, the one who started the competition, sent them a message saying they had achieved absolute model accuracy.
That must have been a really proud moment for the whole team.
It was. For scientists who had worked on this problem for decades, it was a huge breakthrough. And the scale of the achievement is just mind-boggling. In a few months, AlphaFold did what would have taken decades of traditional research.
It's like they sped up science by a factor of 100, and this has had a real impact, right?
Absolutely. It's helping with everything from malaria vaccines to understanding antibiotic resistance to figuring out how protein mutations cause diseases. It's even giving us information about proteins in endangered species.
So a single AI has basically unlocked a whole new world of biological research.
It has. And the AlphaFold paper has been cited thousands of times. It's had a huge influence and, of course, led to the Nobel Prize.
Right, the Nobel Prize. But the prize wasn't just for predicting protein structures. It was also for designing new ones, right?
Yeah, David Baker won the other half of the prize for his work on designing proteins that don't even exist in nature.
Design of new proteins? Wow, that's incredible. How do you even do that?
It's like we're moving from reading the language of life to writing new sentences in it.
I like that analogy. So how does it work?
Well, they're using the same kind of AI that creates images from text prompts. Like those programs that can generate a picture of a cat riding a unicorn.
Oh, yeah, I've seen those.
They're applying those ideas to protein design. Baker's technique is called RF diffusion.
RF diffusion, okay.
Basically, they train the AI to remove noise from protein structures.
Noise, like random distortions.
Yep. So they take a known structure, add noise to it, and the AI learns to get back to the original structure.
I see. So it's like training it to recognize the correct shape, even with all the noise.
Exactly. And once it's trained, you can give it a target function, and it can create a new protein that will fold into the shape you need to achieve that function.
So they can basically design proteins to order. Do you have an example of how they're using this?
A really cool example is with snake venom. You know how they make antivenom?
I don't think I do.
They milk venom from snakes, inject it into animals like horses, and then collect the antibodies from the horse's blood. But this has problems. You can have allergic reactions to the animal antibodies.
Right, I can see that being a problem.
Baker's team used AI to design new antibodies that target snake venom toxins, but are completely compatible with the human immune system.
So a safer antivenom.
Yeah, and potentially much easier to manufacture and transport.
That's a game changer for snake bites. What other applications are there?
Well, they're working on new vaccines for cancer and autoimmune diseases.
That's huge.
And there's exciting work on designing enzymes to capture methane from the atmosphere.
Wow.
And even enzymes that can break down plastic.
So AI could help solve the plastic pollution problem?
It's definitely a possibility. And all this is happening much faster than traditional methods.
I remember you mentioning cowboy biochemistry. What was that about?
Chuckles.
Yeah.
Yeah, that's what they call it in Baker's lab. It used to take months or even years to design a new protein and make it in the lab. Now they can do it in days. It's this super fast cycle of design, build, test. It's a whole new way of doing biochemistry.
So AI is not just making proteins easier to understand, it's actually changing the way they're designed and built.
Absolutely. And it's not just proteins. AI is having a huge impact on other areas of science, like materials science.
How so?
DeepMind has this program called GenoE that has discovered millions of new crystal structures, many of which could be useful materials.
New materials, what kind?
Things like superconductors that can conduct electricity with zero loss,
Wow.
and better batteries for electric vehicles.
So AI is helping us discover entirely new materials that could revolutionize technology.
Exactly. And it's all because AI is helping us solve those fundamental problems that were holding us back for so long.
It's like solving a problem at the root of the tree of knowledge, which then unlocks so many new possibilities.
Exactly. And the speed of this progress is mind-blowing. It's not just making things a little bit faster, it's changing the entire way we approach science.
It's so exciting to think about the future of science with AI. Even with what we have now, the potential is enormous. And as AI keeps developing, who knows what we'll be able to achieve?
I completely agree. We could be looking at cures for diseases, new materials with unbelievable properties, and solutions to environmental problems that seemed impossible before.
So, after this deep dive into AI and protein folding, what's the biggest takeaway for you?
I think it's the amazing interconnectedness of it all. Solving a fundamental problem in biology, using AI, has led to potential solutions for some of the biggest challenges facing humanity. It shows the power of basic science and how it can lead to unexpected and transformative applications.
Absolutely. So, with AI now able to design new things at this speed, what do you think the future holds? What possibilities can you imagine?
It's hard to even grasp the possibilities. But I think an even bigger question is, what new questions should we be asking? With this power, what should we be focusing on? What are the ethical considerations? These are the questions that will shape the future, and I think it's a conversation we all need to be a part of.
Couldn't agree more. It's an exciting time for science, but also a time for thoughtful discussion about the responsible use of these incredible new tools.
Absolutely.
Thanks for joining us for this fascinating conversation. Until next time, keep exploring and keep asking those big questions.
Definitely.
And to all our listeners, we'd love to hear your thoughts. What possibilities stand out to you? What questions are you grappling with? Join the conversation and let's explore the future of science together.